Here is the video lecture that complements your learning of Chapter 6.1 materials. Please pause as many times as you'd like, and especially when I ask you to answer some questions. Here is the list of learning objectives for this portion of Chapter 6. We will discuss these in order. Lipids are macroorganic molecules that are highly hydrophobic. You should know that vegetable oil does not mix with water because of this. Just a semantics here, but scientists use oil to describe lipids that are liquid at room temperature, while fat for those that are solid at room temperature. What are the biological functions of lipids? Lipids are biologically important for many reasons. One of those functions is to serve as energy storage molecules. And also lipids provide insulation for many organisms. Winter survival of many organisms like this bear here depends on the functions of lipids as energy storage molecules and insulation. Also, lipids are important for communication because hormones are modified forms of lipids. Lastly, lipids are the critical component of cell membrane, and we will talk more about this point later on. The basic component of lipid is a fatty acid. A fatty acid is made of a long chain of hydrocarbons with carboxylic functional group at one end. This long chain of hydrocarbon is non-polar and therefore contributes to the property of lipids as a hydrophobic molecules. To form lipids, the fatty acid join with through the process of condensation reaction to a backbone molecule of glycerol. Here is glycerol, and the fatty acids joins with each of those oxygen atom on one end of the chain. The condensation reaction proceeds and therefore forms a covalent bond. The particular name of the linkage here is called ester linkage, and again it is a covalent bond that is formed by sharing of electrons. One property that is unique for the discussion of lipids is the concept of fluidity. Fluidity describes the physical state of lipids as fluid. So increase in fluidity makes the whole structure less rigid. This is particularly important when we talk about the structure of cell membrane. Two intrinsic factors that affect the fluidity of a lipid molecule is the length of the fatty acid chains and the degree of saturation. The degree of saturation measures the number of single and double bonds within a fatty acid. When there are no double bonds within a fatty acid, the fatty acid is said to be saturated. On the other hand, there are at least one double bond within a structure, the fatty acid is said to be unsaturated. So no double bonds means saturated, and at least one double bond translates to say it is unsaturated fatty acid. Here is the chemical structure of a saturated fatty acid, having no double bonds within. Compare this to this chemical structure of unsaturated fatty acid with one double bond in between. Notice that this presence of this double bond introduced the kink in the structure of the whole fatty acid. How does this affect the fluidity of the lipid? We will talk about it in the lecture. So, these are the set of learning objectives that I hope you to have achieved. Please critically evaluate your understanding of each of these items and bring any questions you might have to the class.